you, Adam. That was an excellent talk. So, right, I just recently joined BioTeam, and focus is going to be primarily on helping teams navigate AI. And I have a set of slides here that kind of give a cautionary tale of and describe AI not as a scary monster that I think many of us see displayed, but as a source of great possibility and another tool in your tool belt. So let's define it first, and here's how I see AI. It means using empirical data to generate an algorithm that can predict and make decisions on new data. It is not a black box, as we said before. It can be explainable, and we're working to do better in making it more explainable. As a general, AI is an umbrella term. It includes many aspects of where machines imitate human decisions. Within it, there are traditional statistical methods that we today define as machine learning, and this is generally a computer that's improving with more data. And we're not explicitly programming an algorithm to make a decision. And what has boomed in the last few years has been this new method called deep learning. The math has gotten more available and easier to do because of new hardware. And deep learning has allowed us to look at situations and scenarios where there are many, many, many variables that we cannot account for or possibly create a model for. So what it is, it's a method like any other with inputs and outputs. Somewhere in the middle is the part and the art that we call today doing AI. The results are mathematical. They may replace traditional methods, but they're not a replacement for traditional methods. And I'll explain that a little bit later. And like I said, it's a tool like any other. It is not a black box. Of course, the math that goes in there is very much obfuscated by the methods that were created. But in the end, like any other application, it takes some validation. So a scientist will see the output and validate this output and give it the context that it deserves within the inputs and the outputs that are being within the inputs that are being fed into it. The results are not fact, though. There definitely requires a person that understands the data and understands the limits of the data and the outputs that are coming out. It will probably not replace traditional methods. The idea that this would replace it as a method of understanding traditional methods, for example, doing quantum chemistry, is not going to happen. But it is a method that can get us to answers where perhaps quantum chemistry is still trying to figure out what the actual interactions are. It is generally not difficult to get started. It's getting easier and easier as these new frameworks come along, and I'll show you some ways that you can get started if you haven't already. So when is it appropriate to use AI? Well, I would say start right now. There are lots of ways, and I'll show you in a few slides how you can get started. You may be using it already if you speak to any of these new machines that you can say, hey Siri or okay Google. It's already available there. Lots of our emails are already getting some AI adapted to it to give us contextual information. So you're probably already using AI. In general, though, if you're thinking about applying this in some domain, if time to solution matters, perhaps it's a good motivation to begin looking at AI. And when throughput matters, for example, is another good motivation to think about AI. When you have many covariates to consider, where you don't know what are the things that are affecting all of the one variable that you're trying to look at, this is a good opportunity for AI. When modeling is too difficult or it takes time to develop because we don't understand the phenomenon yet, that's another good opportunity because perhaps you need the answer first before you need to explain why this is happening. And when you have lots of data, it's a good approach today to get your data in ways that are computable. And if you can get to that point, then this is a great opportunity to begin looking at AI. If you don't know where to start in terms of, okay, maybe I've convinced you to use AI, where do I begin? Well, first begin with a specific problem. Not something too broad, like are there aliens out there? That is very broad. I've in my previous role, I walked into lots of researchers and I would ask them, what would you like to do with your data? And they would say things very broad like, well, I'd like to solve cancer. Well, that's very, very big. That's a huge problem to tackle. But can we identify a particular type of cancer in an image? Can I find a dog and an image full of goats? That would be where AI would be very appropriate. There are lots of websites that you can get started. This is actually a website that I've used with my own kids. This is not a toy website. It really does show the basics of AI working, and for folks to play with it, for plays with models that already exist, that people have already put out there. And it gets you a good understanding of what it can and cannot do and how to break it. One example is there were some cameras put out to detect, for example, the license plate on a car. And it could easily identify that also on somebody's dress. So if somebody was wearing a dress with a license plate, it would also identify that as a car. So those sorts of limitations are where these websites make it very relatable to everyday life and how you can break systems like that. Another really great place is this thing called TensorFlow Playground. And this, if you want to get into neural networks, which is where it lives within deep learning, which is where folks that have a lot of different variables want to dip into, this is a good place to play with the kinds of methods, that math in the middle that I mentioned could be obfuscated to most of us. But it does describe the sorts of inputs that go in, the sorts of decisions that are made within the network, within the neural network, and the outputs that then come out of it, and the sorts of cause and effect that can happen when you put it in there. Another great place to do some learning is this particular blog here. Andrej has been able to really concisely describe the great pieces and algorithms and methods and neural networks. And I highly recommend giving this a read to kind of give you a broad sense of what you can do. On the data side, assuming you're ready to do AI and you say, okay, well, what should I expect? I think the main thing is to just accept now that your data will be bad, and that's okay. You don't have to have clean data to get started. You do have to have data that's good, and that's where you're probably going to spend most of your time in. Your data will likely have artifacts. It'll have characters that you don't expect are there. It will be incomplete. It will be skewed. It would be either over-representative of one type of scenario versus another. It'll likely be biased. We all have biases ourselves. So you can think in the sense of medical data, for example, there are biases that go into the medical data and how certain groups and categories get treated. For example, children. There may be some assumptions about children, the kinds of drugs that children are given, because some drugs are not tested in young children, so they may be given other kinds of drugs. So all of these biases will go into the data that you're looking at. Chances are your data will be wrong. And more data sometimes help, unless it's more wrong data. But again, you're going to spend most of your time actually analyzing the input. It'll probably multimodal. That means that you're now not just looking, if you're looking to do AI, you're probably not looking at a simple regression. You're not looking at a simple variable. You're probably looking at combining different kinds of data sets. So lately in the healthcare realm has been a lot of, I would like to combine genomic data with other kinds of results. For example, let's say hospital results. So that would be a good opportunity to think about AI. And very much noisy. Data is noisy from lots and lots of different inputs where humans are involved, and even sometimes when humans are not involved. So there's only so much you can do to reduce the noise. But this is part of AI. Your data and how good it is part of AI. Embrace it and just accept before you get into it that it's going to be bad, but you can make it better. I have an example here from a longtime collaborator. We're actually co-authors way back when I started my career about 12 years ago. These folks, and here's an example of just sort of a fluke idea. In Scotland, what they have is they do these kinds of infrared testing. They basically put a piece of light through the milk that is collected from cows to determine things like protein content and fat content, et cetera. And so somebody one day decided they had a question: can I determine whether or not a cow is going to be pregnant by just looking at this kind of data? So this is instrument data. And lo and behold, it's exactly what they were able to find. They were able to determine pregnancy. And if you know anything about farming, one way to determine pregnancy is to have a vet come in and palpate the inside of the uterus of the cow. So by then, there's quite a large fetus in there for them to be able to feel it. And that's expensive, time-consuming. And instead, perhaps you can figure out if your artificial insemination, another kind of AI, has actually worked. You can reduce the time for the next impregnation to be able to continue making milk. So for them, this is going live countrywide and may actually go live for the whole of UK, and other places are looking to take it. And it just started with a question: can we make this happen? And the one a conversation I had with this collaborator was they weren't sure how to use the data, and the little piece of advice I gave him changed the game, which is a lot of people out there take instrument data and turn it into images, and then use those as images to then do the kind of analysis that he was looking at. And that made it so much easier. So the data side actually decreased quite a bit because they were able to use images instead of the raw signal. So that's a little bit of an example of sort of an everyday life, a small question. This whole project took about 6 months to get started, and the last 3 months was just fine-tuning it to get the accuracy up. And in the side of drug discovery, if you are in the realm of drug discovery, it's a hard slog to do drug discovery. I think everybody understands that it takes several years, sometimes decades, to go through it. It's very expensive. But we do have today a large corpus of data that includes whether or not these drugs have failed, the reasons why they have failed, and especially if they've gone through trial, and understanding whether or not they failed and or succeeded because of they weren't therapeutic or they were actually harmful. And that's very valuable data today to mine. If I can find out if a particular drug is harmful because it has some side effects, and it seems like a class of drugs end up having the same side effects, something like deep learning can tell you right away if this new drug you're looking at, or this new molecule that you might be considering, is going to create those sorts of side effects down the road. So you're not spending 5 years trying to do assays and every other piece of the process before you actually go into, phase trials. So molecular docking is the beginning of it. Can I fit it in a certain pocket? This is very important, for example, in antibiotic discovery. We know antibiotics are very difficult to find, and often the bacteria themselves are mutating. And there's only really a certain number of classes of antibiotics that we have today. People can be allergic to a whole class, for example, like penicillin, right? And so they can't use that particular class of drugs. So it's very important for us to be able to, adjust to the mutation rates and not have to have that large, long slog of a process of discovering new drugs. Molecular docking, though, takes a long time. It takes minutes, sometimes days, depending on what you're modeling. Just to sample the space of fitting that molecule in this pocket and rotating it and turning it around. You may or may not even have a good idea what the target site might be, and you may or may not have a good idea what the actual protein looks like. And those scenarios where you don't have enough information about the problem you're trying to solve is where AI actually becomes very useful, provided obviously you have appropriate data. So the next talk we're going to hear is exactly that. It's solving a problem of finding a new drug from drugs that we may have already come across. And in preparation for this talk, I wanted to understand more about the work that John Stokes had done, and his team. So I went and played a little bit with this framework called ChemProp. It was developed by MIT's CSAIL. And what it does is it's a neural network specifically for drug discovery. And again, it has inputs and outputs, and the input is very simple. The encoding for the data is encoding SMILE strings for a particular activity It's a binary activity, so 0 or 1. Does it work? We know that this drug works for this particular feature that I'm trying to identify. So it's a very simple input. The neural network has been used before. It's actually been used before in another context. So here's a paper from where it was first introduced, and it's called the message passing neural network. And you can see what we were talking about before, how, for example, DFT, which is a type of molecular simulation, quantum chemistry, may take quite a few more magnitudes of time compared to, if you can get that data and train it and then push it through this neural network. This is an excellent paper. I definitely encourage you to read it if you're interested into how these networks are built. But this is what was used within ChemProp. And so I was able to do a little study of that. And what was really nice too was that the folks that developed ChemProp actually developed a web interface. And so you were able to upload your data to train it. There are already pre-built models they had done. And the models are available, including the model that for the talk you'll see after this one. And it was pretty easy to upload some data, a short set of smile strings and a short set of whether or not they are active for that particular feature that the model was trained for, and then and get an output. And I just happened that that day I had taken my asthma drug, which is called montelukast, and I decided on a whim, because that's exactly what was on my mind and on my desk, to put it in there. And I got a number. I got 0.60, and I said, well, what does 0.60 mean? What is this output? It's an activity probability. And I said, okay, so what are other things that are known to be active in this scenario? And so this is a particular model they had trained for SARS-CoV for a particular protein called 3CL-Pro, the 3-cytokine-like protease. And when I searched the literature, the other things that were supposed to be as active or have a high probability actually came in lower than this drug. And then I tested another drug within the same family, which is a leukotriene receptor agonist, zafirlukast, and they came up with an even higher probability. And so, figuring this was a fluke, I decided to prove myself wrong. Now, I am no expert in any of these drug families. I am not a drug discoverer or researcher, but I do have friends that are in that realm, and they thought that maybe this was a curious thing that warranted taking a deeper look at. And so they've taken these very playful results, this little exercise that I did to just play with the framework. And they've taken these and presumably they're doing something within assays that are much more rigorous than me playing with a framework. But this is a sort of, fresh kind of, it gets you an idea, gets you something going, gets you researching. And that's really the promise of what machine learning AI can do, is get you some new places to look where perhaps nobody else is looking at this point right now, and give you new ideas. It's another tool in the tool belt. It does not eliminate the tools of traditional drug discovery. And so with that, I'd like to invite you to come talk to us at BioTeam about your scientific goals. I failed to put my email here, but you can reach me at fernanda@bioteam.net. Thanks.